Hello friends, welcome to CEC Live Lectures. Dear friends, today in this session we are going to continue further in our series on principles of ecology. Dear friends, this is the second lecture in this series and today we are going to talk on population ecology. Friends, we are going to have more and more session on population ecology. This is the first session and for the discussion we have with us in our studios Dr. Charu Dogra Rawat. Dr. Charu Dogra Rawat is Assistant Professor in Department of Zoology, Ramjas College, University of Delhi. Dr. Rawat is a prolific professor and through her we always get in-depth knowledge on various topics of zoology. Friends, if you wish to ask questions from Dr. Charu Dogra Rawat, do call us through our toll-free number. Our number is 1800110430. I repeat, our number is 1800110430. You are requested to call in the last 10 minutes of the lecture only, as well as please ask questions relevant to the topic only. Now, I would like to welcome our guest, Dr. Charu Dogra Rawat, once again. Hello, ma'am. Welcome to the session.
So, starting with the population ecology today, which is a very important aspect to learn more about the principles of ecology. In this particular session, our learning objectives would be to understand the definition of population, what actually this population word means. We'll also understand the aims of studying population ecology. Why do we have to study the population ecology? We'll understand about the spatial structure of a population, as well as we'll talk about the age structure of a population. So this will be the talk for the today and covering all these topics. So starting with, what do you mean by population? A population is a group of individuals of the same species that inhabit a given area. Now there are two important points to note for this definition. One feature is that the individual of the same species, which means that potentially, usually in sexually reproducing organisms, they have the potential for interbreeding among the members of the same population. So what we generally refer to as the gene pool. So when the same species, the individuals of the species can interbreed with each other, they share a common gene pool, and that gene pool basically forms the set of a population. The second important feature is the spatial concept. When we talk about that, it requires a defined spatial boundary. It's in a given area. The individuals of a given area are termed as the population. So one thing is that they are freely able to reproduce amongst each other, and second thing is that they are confined in a particular geographical region. So example is the population of Darwin's ground finch inhabiting the island of Daphnia Major. We have heard about Darwin, we know about the Galapagos Islands. So the different species of finches which he found. So for example, one of the finches, the ground finch, the large ground finch that inhabit that island will constitute a population because it's in a defined area, as well as the ground finches can sexually reproduce amongst each other. Now, threefold aims of population ecology we will like to go through before we start with this, because we must have an idea why we are dealing with this or why we do we have to study this at all. The first and important population ecology aim is to elucidate general principles explaining dynamic patterns of species abundance over time, including relative constancy, cycles, irregular fluctuations, violent outbreaks and extinctions. When the species are coexisting in an environment, or when the individuals of a particular species are living in an area, they are not constant. It's a very dynamic in nature. They keep on increasing in number, or their numbers keep on fluctuating because of certain features such as extinctions, or there are certain violent outbreaks that occur. So to understand all these dynamic patterns of species abundance over time, we need to study the population ecology. Second aim is to integrate these principles with the mechanistic models and evolutionary interpretation of an individual, life history tactics, physiology, and behavior, as well as with theories of community and ecosystem dynamics, which basically means that whatever we come to know about the principles of the different kind of developments taking place with the individuals, we can now simulate them into certain mechanistic models, and we can make evolutionary interpretations about the individual life history, tactics, behavior, physiology, etc. As well as we can study the theory of community and ecosystemic dynamics. And third is to apply these principles to the management and ultimately the conservation of natural populations. So we study populations so we can understand the existence of population, we can understand the dynamics of the changes that they are occurring in a population. We can understand the or we can simulate certain models related to those principles, and then finally we can devise strategies and formulate principles so as to manage as well as conserve the natural populations. So these are the aims of studying the population ecology. Now when we talk about population, we talked about two aspects. One aspect is that it is composing of individuals. Now what constitutes an individual? In most animals, the life cycle starts with a fertilized egg, passes through a largely irreversible process of coupled growth and differentiation, and ends in a tightly integrated, what is called as a unitary adult organism. So that is what we understand as an individual, for example, when we talk of humans or when we talk of any other animals. So in most of those animals, the existence is a unitary adult organism. So for us and for ecologists, that constitutes an individual. Form, development, growth and longevity of unitary organisms are predictable and determinate right from the conception on. So we can just find out these features, and it's easy to predict all these features and determine all these features, such as how will they develop, what kind of growth will they attain, how much will be the average longevity. So we can easily predict that, and it's basically specific right from the when the conception is started. So for a unitary organism, that constitutes an individual. But in most of the plants and some colonial animals such as sponges, growth and differentiation proceeds in a modular fashion. The growth involves replication of a basic body unit so that a fertilized egg generates a spatially distributed population of connected modules, which basically refers to a more modular kind of organisms. The modular kind of organisms basically means that in some way they are connected to each other. These modules, the population of modules which are generated from a single organism, they are connected to each other, and this kind of growth involves replication of a basic body unit, and the whatever the offsprings are there, they are connected by certain things. So they are connected modules which are there. The connection between these modules sometimes break, leading to a dispersed clone of physiologically independent units. So whatever they are producing, so at first by sexual reproduction, one of the organism is formed which is termed as a gamete, and then these gamete will give rise to the clonal population which will be connected to each other in some manner. And these things are then called as the kind of a modular organism. We'll take an example, for example, aspen tree, which is Populus trameloides. Growth involves modules of roots and root buds which give rise to the clones. So you can see the younger ones are the smaller, which are farther away. So you can see that there is a parent on the right hand most side, and this parent will be formed by the sexual reproduction, and then their roots will be connected to the younger one, and they will form by the asexual reproduction. And as you can see, through the roots and the root buds, they are all connected. So these rametes are the clonal propagation of the same parent, but they keep on forming. So we as a biologist, this entire thing will be one organism, because it's just one gene pool. But for an ecologist, they are the clonal population, so it's just one organism. But for an ecologist, each tree, each trunk can constitute a separate population. So these ramets, what they are called as the clonals, they can be separated from the gamete, or they can just be associated also. So this is one example of a modular organism. So the individual is again, the defining an individual depends upon the kind of study you are doing and the kind of organism one is dealing with. So we can define them in the unitary or in the modular manner. Now, what defines the spatial location? Now, another feature of the population is that it has to be in a particular defined area. So, this spatial location, how do we define it? Spatial location, that is the area over which a population occurs, is defined by its distribution, by the individual's distribution. For example, when we see these red dots represent individuals, and these red dots are lying at certain distances from each other. So some are like accumulated, some are separated. So these are all individuals of a population. Now, when they are enclosed in a particular area which is represented by this blue line, then this is defined as one population containing the individuals of the same species and enclosed in an area. So this particular blue thing is a geographical range. It might not necessarily be such defined thing, but the geographical range is the area that encompasses the population of a species. So when we talk about spatial location, there has to be a certain geographical range which is there. There must be an area where the population or the individuals of a population of the same species are residing. Distribution of a population is influenced by occurrence of suitable environmental conditions. Now, how do we define this geographical range? So, for the understanding of the delineation of this geographical range, or the kind of the places which these individuals occupy, is generally governed by various factors, and one of an important factor is the suitable environmental conditions. So, for example, if we looked at the distribution of red maple Acer rubrum, you will see that the distribution is just like this. It is restricted to this part of the United States, where on the northern side it is the limit coincides with the area in southwestern Canada where minimum winter temperature dropped to -40 degrees Celsius. So the geographical range of this particular, the Acer rubrum, is defined by the environmental conditions. It does not go beyond this, because this is the boundary where the southwestern Canada starts and the minimum winter temperature drops to 40 degrees Celsius. Southern limit is the Gulf Coast and the southern Florida. Dry conditions halt its westward range, so it does not go beyond this. The geographical range for this particular species restricts to the part where it is a little wet. It does not move into the dry conditions which halt its westward range. Within this range, however, within this particular area, the greener area, it thrives on a wide range of soil types, texture, moisture, acidity and elevation. So the geographical range is defined where the individuals of a population reside, and this delineation of the geographical range depends upon a lot of environmental factors, as we have seen. Now, within this geographical range, it might be able to survive on variety of textures, soils, elevations, such as, for example, the red maple tree. However, certain will be further restricted by the environmental conditions, again within the geographical range also. So the distribution or the spatial location is an important aspect to understand when we deal with the population ecology. One important thing which we saw here was the role of the geographic barriers. The southern and eastern limits to its distribution correspond to the Atlantic coastline. As we saw here, the southern and the eastern limits was limited basically to this Atlantic coastline. So these trees might have suitable environmental conditions in Europe or in Asia, but they are restricted to this particular geographical range because of this geographical barrier, the Atlantic coastline, which they cannot cross and go to the European or the Asian countries. So such is the role. So when we talk about dispersion, we'll talk about in detail about such geographical barriers. But again, the distribution of a particular species or a population is affected by such geographical barriers. Other barriers such as mountain ranges or extensive areas of unsuitable habitat may likewise function to restrict the spread and therefore the geographic distribution of a species. So it is very important to understand that the population is defined in a geographical range, and that geographical range is affected by various factors, various geographic barriers, and within that particular geographical range, the individuals of the same species are referred to as the population, which we talk about. Now, the distribution of a population can be described at a number of spatial scales. So, this is again one of the concepts which is important to understand, that when we are talking about a distribution of population, we can actually define this distribution at different scale levels. For example, if we are talking about in the at the world level, then the distribution of a population can be described in a different manner when we compare the distribution of a particular species among continents. But when we compare the distribution of a particular population within a local area or within a regional area, then the distribution can be described based upon the local factors or local climatic things also. So we'll take an example here. For example, this is an example of a distribution of the moss, which is termed as Tetraphis pellucida, from its global geographical range to the location of individual colonies on a tree stump. So this is basically the geographical range or the distribution which is confined to the North America for this particular moss. So as you will see in the North America, then it is confined to this particular region. So within the continental, so this is the worldwide distribution what we are talking about. This is the continental distribution we are talking about. Then in the constitutional, in the continental distribution, when we look at the regional level, then again there is a different kind of distribution which we are talking about here. Then when from the regional level, if we go to the clusters, we look at that, then we will see that these clusters are confined to the coastal basins. When we further zoom it, we will see that in these particular local regions, they are then confined to certain stumps only. So you will see that and they will correlate with the conifer trees that exist here. So they are just confined to that area. So in here, these are the coniferous stump and they are just restricted here. And in fact, if you look at the stump, then they will form clumps in such a manner. So, when we talk about the distribution of particular individuals or distribution of a population, we can describe it at all the spatial scales. We can talk about in the terms of worldwide manner, we can talk about in the terms of continental manner, we can talk about it in the terms of, and we will see that all these, spatial distribution is correlated with certain habitat that it requires or certain environmental factors which it is receiving. So, this particular distribution of the moss from its global geographic range to the location of individual colonies on a tree stump very well tells that the distribution can occur at all the scales. At continental scale, suitability of climate is the factor. For example, temperature and humidity. So the more suiting temperature and humidity is in the North America, and that too towards the eastern side. In an aerial, when we talk about a regional area, then the microclimates along the stream banks where conifer trees are abundant, that is where this particular moss is going to be distributed. Then within a locality, for example, when we talk about these stumps, then the stumps of the trees where pH is sufficiently acidic, they will colonize. As you can see, they have colonized here because there might be a sufficiently acidic environment. So as I said, spatial distribution will depend upon the lot of factors, the environmental factors, the habitat, the choice, preference or suitability as we call it, then different conditions, which will assign the population or the individuals to different regions. So that is how the population is distributed. Then there is a concept of what is called as metapopulation. So before we go into detail about talking about details of the population ecology, we must understand the different terms. So metapopulation, when we talk about that, it is a collection of local subpopulations. So subpopulation exists because of the environmental heterogeneity in a particular geographical range. Within a particular geographical range also, the environment might be different. There must be patches where the environment is favorable, but there might be patches where the environment or the habitat is not suitable or not favorite. So the individuals or the population is going to be, they are going to be clustered in those areas where the environment suitability is there or where it can tolerate its existence, external environment. Therefore, there exist these kind of local subpopulations. Ecologists call the collection of these local subpopulation as a metapopulation. An important aspect of metapopulation is that there has to be some kind of exchange, some kind of reproductive exchange that need to occur between these patches. Then there is not a random panmixia, but even if there is no interbreeding which is going on, then not necessarily it will be a part of a metapopulation. So the important point is that based upon the environmental heterogeneity, local subpopulations will constitute together as a metapopulation, and the requirement is that there must be certain kind of interbreeding or certain kind of interaction between these subpopulations which is there. The metapopulation concept was first introduced by ecologist Richard Levins in 1969 and further developed by Ilka Hensky and among others. So here you can see these are the patches which are occupied, and there is a movement and there is an interaction that can occur between these subpopulations. This is a particular patch which is right now empty but can be taken. So it can be, there is a suitable environment, but right now the individuals have not transversed to that particular patch. So such is the concept of metapopulation, and we will deal with different kind of metapopulation dynamics when we talk in detail about that part. Coming to now the features or the attributes of population. One important attribute of population is the density. So density is the crude density, what we are going to define as the number of individuals per unit area, which is the per square kilometer, hectare or per square meter. So that is what is called as the density or crude density. So density basically means that within this geographical region, within this geographical area, how many number of individuals are present. And these number of individuals, when we divide by per unit area, we get the density, which is the crude density. The calculation is by, for example, if we divide it into a grid sort of a thing and we calculate that the region 1 actually has 1, 2, 3, 4, and this grid is, for example, a 1 by 1 meter square. It doesn't look like that, but it is a 1 by 1 meter square. So there are 4 individuals per meter square. That is the crude density. In the region 2, there is 2 individuals per meter square. And in the region 3, there is 0 individuals per meter square. So this represents the density or the crude density. Now this does not represent probably the density of the entire population, but we are right now talking about the density at per square kilometer or per unit area we are talking about. So if we consider this as one unit area, then we just count the number of individuals and we just say that 4 individuals per meter square is the density or crude density of this particular population. But there are patterns of spatial distribution, and the spatial distribution can be of 3 types. One, it can be clumped. So there are certain regions where the number of individuals are found more. Again, as we said, it will depend upon the microclimate or micro environment which is found at the certain areas. And then the spatial distribution can also be random, where they are just occupying random areas. Or the spatial distribution can be uniform. When we talk about uniform distribution, then there is equal distances between the individuals of a population. So spatial distribution, this is the most common type, the clumped spatial distribution, but the individuals of a population can be distributed in either of the manners. In the random dispersion, if the position of each is independent of each other, so there is no correlation between the distribution, they are just lying randomly in a given geographical range. Uniform dispersion if there is results from negative interaction such as competition among individuals, or what a concept which is called as territoriality, which is there is a favorite habitat which is there, or there is some kind of regional binding which is there, or if there is a negative competition, then they are going to occupy such certain areas only, and they will have an equal distance between them, and therefore will have a uniform distribution. Clumped distribution is the results from a suitable habitat, resources distributed as patches, or there are social groups, economics groups, or geographical groups as found in case of humans. So the patterns of spatial distribution can vary, and accordingly the calculations of density will also vary, which we'll talk about. Thank you.
With this note, thank you, ma'am. Thank you so much for giving us this session. Friends, you are requested to be with us as we are back after a short break. Thank you. Hello friends, welcome back to this session. Friends, in this session once again we are going to talk more on population ecology, and for the discussion on the topic we have with us in our studios Dr. Charu Rawat. Dr. Charu Rawat is Assistant Professor in Department of Zoology, Ramjas College, University of Delhi. Friends, if you wish to ask questions from Dr. Rawat, do call us through our toll-free number. Our number is 1800110430. Now I would like to welcome our guest Dr. Charu Rawat once again. Hello ma'am. Welcome back to the session.
Now, density of a population is defined as the number of individuals per unit area. So, we have been talking about this density, which is actually the crude density. Now, we have also seen that the spatial distribution of individuals is not just uniform. It can be uniform, but it can be random, or it can be clumped depending upon the availability of resources, or when they do not have to interact with each other at all. So, these kind of spatial distribution makes it difficult for us to ascertain the density of a population by the definition of the crude density, because if we, for example, in this particular case, if we divide it into equally grids, at one particular place we are getting the density to be 4 individuals per meter square, whereas in the other area we are getting 0 individuals per meter square. And the most common form of, or the pattern of spatial distribution is the clumped distribution. So, in the clumped distribution or in random distribution, it becomes very difficult to ascertain the crude density, and it is actually not the true reflection of the number of individuals in a population. Usually, for such a place, what we talk about is what is called as the ecological density. Ecological density is the number of individuals per unit of an available living space. So, for example, we were talking about the moss tetraphis, then tetraphis is confined to the area where the coniferous trees are there. So, the resources is there, that's a clump division. So, we do not take the entire geographical region, but we take the available living space, which is that particular area, and we talk about the density, then it is termed as the ecological density. This accounts for the patchiness. However, this ecological density is very rarely estimated because it is very difficult to ascertain the number of individuals, or it's a very hard task to just ascertain the number of individuals in a particular area and take it as the density of a population. In a study of bobwhite quail, for example, Colinus virginianus in Wisconsin, density is expressed in terms of number of birds per mile of hedgerow. So, it is not defined as the number of individuals found in Wisconsin, but it is found correlated with the availability of its specific or preferred habitat, which is the hedgerow, and then the density is ascertained. So, such a kind of a density is termed as ecological density. Now, when we talk about abundance, it reflects both the population density as well as distribution. So, while talking about the spatial structure of a population, we have talked about the distribution as well as the density. Now, the abundance reflects or covers both the features, and therefore, abundance defines the size of a population, the number of individuals in the population. So, our aim was basically in the starting to understand the abundance part, that how many individuals are present in a particular population, and that we can understand only by understanding the density as well as the distribution of individuals within a geographical range. So, abundance or the population size is given by density multiplied by the area it covers. Now, an important aspect to know about the abundance of a population is to measure the density. Now, measuring the density of individuals or unitary organisms or particular organisms, which are very rare or very less in number, for example, endangered species or extinct or on the verge of extinction species, it's relatively easy because at that point we can just count the number of individuals, when the density is low or when the number of individuals. So, one method is the counting method, when both the distribution and abundance are small, usually of the rare or endangered species, as we have said, or in usually open habitats. For example, if we want to know the density of antelopes living on an open plain or waterfowl concentrated in a marsh, then in such an open plain, it's easier to just count the number of individuals and measure the density. However, in all others, in most of the cases, density is estimated by what is called as sampling the population. So, you cannot count individually all the members of the population. However, what you adopt is a method what is called as the sampling of the population. So, sampling a population basically means that the part of the population that is actually observed forms a sample. So, you do not actually count the number of individuals of an entire population, but you count only a part of the population, or you ascertain the density only in a part of the population, and that part of the population which is actually observed is termed as a sample. The sample of objects, however, should be representative of the entire population. So, this choice is very, very important, because whatever sample we are considering, it should be representing the entire population. A sampling procedure that assumes that each object in the populations has equal chance of being selected is termed as simple random sampling. That is the most commonest form of sampling which is used for measuring the density, and that simplest form of sampling is what is called as the simple random sampling, that within a population randomly you take out or you count, or individuals you randomly take out samples which basically reflects that every individual of the population, usually we do not call them, as ecologists do do not prefer to call them as individuals, but they call them as objects, because individual, it just can be a leaf or it can be a stem of a tree when we are talking about the population. So it generally refers to it as object. So if we are referring to the object in the population, then the chance of getting selected, when the equal chance of getting selected each individual has or object has, then it is termed as the simple random sampling. There can be stratified random sampling, where the random samples are taken from each of the defined subpopulation or groups. So as we have seen that, the preference of or the occurrence of subpopulation or the restriction of subpopulation depends upon a lot of factors. There are certain environmental factors, or as in the case of moss, there was the presence of the coniferous trees along the streamline, the stream or the banks of the stream, it was found there. So the stratified random sampling is that from each of the subpopulation, if we take random samples, so that becomes stratified random sampling. Sometimes what we want to know is what is called as a systematic random sampling, and systematic random sampling refers to the fact if we are interested in systematic variations within the population, usually along some gradient. For example, if we are looking at a fish which is found in the deep water or in the surface water and there is a variation in a size. If we want to understand the variation in a size, and if we just take the random samples, we have to make a systematic random sampling, because we want to know how many larger fish are there. So we must collect from that area where the larger fishes are present, and we must report that okay, they are found in the depth area or they are found in the surface area. So this becomes a systematic random sampling when we are generally studying a particular variation or particular feature which is occurring inside in a population. So these are the different kind of sampling we do. Now there are different techniques that how do we measure density. So these were the kind of sampling we were doing. Now coming to the facts that how do we calculate the density. An important method of calculation of density is what is called as the quadrates method or sample plots. So you can see the grid which is lying at the back, that is the kind of quadrates which we put for the sampling. It is used widely in the study of population of plants and sessile animals, or the animals which do not move. The area of the study is divided into subunits, individuals counted in only a subset or sample of the subunits. So as we had seen in the example, in a hypothetical example, when we talked about the geographical range, we just put a grid over it, and from that grid we just take random samples 1, 2 and 3, and there was a whole variation. In one of the grid there was 4 individuals per meter square, in another grid there were 2 individuals, and in another grid there was 0 individuals per meter square. So we randomly sample the subunits, we count the individuals, and then we take the average or the mean. So that is a quadrate or a sample plot method. From these data, the mean or the average density of the unit sampled is determined. Accuracy of sampling can be influenced by spatial distribution of individuals. So we have been repeating this because it depends upon what kind of distribution is there, a uniform, random or clumped distribution. So obviously the method of sampling will differ. In clumped, in uniform, it will be all equal. From every quadrate you will get an equal number of density. So average will be the same. But in case of random, or more so in case of clump, you will have a variation higher and lower, but the average should be or the mean value is what we generally consider. But accuracy of the sampling will definitely be influenced by such kind of a spatial distribution. It can also be influenced by the choice of boundaries or sample units. So whatever grid we are choosing or whatever grid we are keeping, then this particular sampling method will also be influenced by the choice of such sample units which is there. So to understand or have the true reflection of the measure of density, we must consider the estimate of variation, or we must provide a confidence interval for the estimate of density. Now what this variation and confidence interval means, we'll talk in detail when we talk about the quantifying ecology or the statistics related to it. because the variation basically means that when we have an average, a mean value, then on either side they vary, there is a kind of a standard deviation which is found. So I'll have a separate session on the statistical or quantifying ecology, and then we'll discuss in detail these terms. But right now at this point I must emphasize on the fact that when you are measuring density, particularly in clumped regions or particularly in regions which has random spatial distribution, then along with the density or the mean density, we must have the measure of the estimation of variation, or we must project the confidence intervals for the estimation of density which is there. Where clumping is a result of habitat heterogeneity, index of ecological density is used. So as we have already talked about, the ecological density is basically the we calculate which is not per unit area but per unit available space, or per unit, for example, the habitat only is heterogeneous in case there are certain suitable habitat regions and then there are unsuitable habitat regions. So in that cases we do not consider the number of individuals in the entire geographical area, but we will consider the number of individuals in a particular suitable habitat area only, and that will refer or give us the index of the ecological density, and that should be counted when there is a habitat heterogeneity which is found. Then after the quadrates method, there is one method which is called as the transects method. Transects method is useful when the area to be sampled is zoned or has some sort of gradients running through it. In transect method, usually what we have, there are two diagrams which is shown here. So transect lines you can see. So what happens is that you define a particular line in this area and then you start measuring across it. If you measure across this particular, if you just sample across this particular line, then that is said to be as line intercept. If you move to certain areas, up till these particular areas, then it is said to be a belt transect method. But then the entire geographical region is divided by these lines, and then the random sampling is done along these transect lines. So that is also one of the method. So sampling is done, random sampling, and can be done simple random, systematic random, or the stratified random sampling. It can be done either ways, but this is just a technique to measure the density. So we measure along these transects line and then calculate the density or the mean density which is there. Now for the mobile populations, an important method is what is called as a mark recapture method. So in this method, the individuals are captured, marked, released, and then recaptured. So the assumption behind mark recapture method is that the proportion of marked individuals recaptured in the second sample represents the proportion of marked individuals in the population as a whole. So this is an important assumption which this mark recapture method makes, but it is the most prevalent method to calculate the density of the mobile populations. So what happens in here is that in algebraic terms, you can see that there is a correlation between the animals which are recaptured on the second day and the size of the sample divided by the size of the sample on the second day, which is equal to the animals marked and released divided by the population size. So we know that how many animals are marked and released, we know the animals which are recaptured on the second day, and we as well know the size of the sample on the second day. So by knowing all these three terms, we can actually predict what is the population size, and this was given by what is termed as the Lincoln-Peterson index. The relationship is referred to as the Lincoln-Peterson index, where n is the population size estimate, m is the marked individuals released, S is the size of the second sample divided by R is the marked animals recaptured. So obviously it makes a lot of assumptions that the marked individuals, they just go and they remain, they do not die, and they just go and mix in the population uniformly. So all these assumptions are there, but it is a very prevalent or accurate, most accurate method for counting the or for finding out the density of the mobile animals which is there. Now, what I have to talk about is the indices of abundance, which basically means that to find out about the abundance of particular individuals in a population, we need not actually count them, but there are certain attributes which are considered, or certain indices which are considered, and they are taken to be as the indices of abundance. For example, methods that relate to the presence of individuals rather than the direct count of the individual. So, when we consider the vocalization, the counts of vocalization, so of a particular individual, we can hear the sound and we can record the sounding. So, the number of times we hear the soundings across a particular area, moving in a transect line, for example. So, we can ascertain the density of that particular individual based upon the presence of the individual, which is conferred by the vocalization rather than the direct seeing or looking or observing and counting. So, it is most prevalent in case of wild animals and wild birds. Counts of animals scat seen along a length of road which is less travelled, or the number of animal tracks that can be seen or found. So, all these things, these are the indices of abundance. They are not the direct count of the individuals, but they indicate that the such and such or number of organisms are present. A series of such index figures collected from the same area over a period of years depicts the trends in abundance. So, only one particular count will not be a true measure of the density, but in a series of such indices or figures collected from the same area over a period of year might depict the trends in the abundance. So, this is also an important feature of calculating the density which is there. So, we have talked about the spatial structure of a population, and in that spatial structure we have basically talked about the spatial distribution of the individual. So, individuals we refer to as the we can based upon the study we are doing or based upon the organisms we are dealing with, they can be a unitary or a modular organism, and that unitary or modular organism are then distributed, or they must be confined to a particular geographical range. So, they are potentially interbreeding and they are defined under in a particular geographical range, and the spatial distribution within those can differ. There are different patterns. It can be random, it can be uniform, or it can be clumped distribution that occurs. And when we are talking about two important attributes of population, that is calculating the density, we must consider the spatial distribution which is there. So, right now we have only talked about density, we have talked about crude and ecological density, and we have talked about how to measure this crude and ecological density, which is basically referring to the number of individuals in a particular given area. Now, but the number of individuals does not necessarily mean that you have to directly count the number of individuals. It's easier to count the less number of individuals if they are present, the individual which are endangered or which are rare, or such individuals can be actually directly counted. But for most of the animals, we cannot count the number of individuals present in an entire population. So, what we do is we do sampling. This sampling, preferred one is the random sampling, and depending upon your need it can be stratified and systematic. So, when we do the sampling, we actually take a part of the population, and this part of the population is actually representing the entire population. And from this part of the population we make predictions about the density or abundance of the population of the individuals in that particular population. So, then we talked about the measuring this spatial distribution, and the measure about, or the density which we talk about, we have to measure by using different techniques, and we have talked about the techniques such as quadrats, transects method, or we have talked about the different kinds of sampling when we do, and then we adopt certain techniques and methods to do the counting of the individuals. And we have also talked about certain indices which only tells about the presence of those individuals and not actual counting. So, this entire constitutes the spatial structure of a population. Now, coming to the age structure of a population. So, now as a population can have a spatial structure because of differences in distribution it does have the difference in the ages of the individuals. If all the individuals had to be conceived at the same time, and they have to be, growing according to the same pattern, and they are just conceived, gave birth, all is happening at the same time, then we do not deal with this particular attribute. But it's not like that. There are individuals which are present at different ages, and therefore the population, we also need to study about the age structure. The number or proportion of individuals in a different age class reflects the age structure. The population can be divided into three ecologically important age classes or stages: the pre-reproductive stage, the reproductive stage, and the post-reproductive stage. So most of the populations the ecologists divide into these three important classes or stages. So pre-reproductive stage, which is sometimes, for example, in humans it's called as the juvenile or the young stage, then the reproductive stage or the adult stage, and the post-reproductive or the old age, as we talk about. So these are the proportion. Then there are different proportion of individuals in all these stages or classes. Determining the age structure of a population requires some means of obtaining the age of the members of a population. Now, when we are talking about the age of a population, and we actually have to find out the age of the individuals. So different means or different ways are adopted for counting the age of the different individuals. If, as I said, they have been born on the same time, it would have been easier to predict the age of the individuals. But since they are different aged, all present at the same time, one has to ascertain the age of the individuals to predict the age of the population. Certain methods in case of birds and mammals are like this. For example, in case of leading primary feather on turkey. So for the adults it's more rounded, whereas in juvenile stage it is more pointed. So one can find out the age of the turkey by looking at the leading primary feather. Similarly, the bone structure in the wings of the bats. So you can see the bone structure. In the juvenile, it's like this. In the adult, it is a little gap is there. So the bone structure differs in the case of neonatal, juvenile or adult bats, and this can be observed by pressing the wings. Just by feeling, it can be ascertained whether what kind of bone structure it has, and therefore then the age of the bat can be determined. Then for plants, it's very common that the cross section of tree trunk showing annual growth rings. By measuring the width of each ring, a pattern of radial growth through time can be established. So for timber, it's very common that you create a cross section and you find these concentric rings which are there, and by the width of these rings, when we can actually calculate the age of this particular tree, as well as the difference between the width as well as the healthiness of these ring areas can also help us decipher the climatic changes this plant has gone through. So certain areas are when there is a lot of water, but then when the rings actually shrink a lot, then there is a scarcity of water or there might have been drought. So ecologists make a lot of predictions based upon that, but age definitely can be ascertained by measuring the width of these rings. So the age can be determined in that particular manner. Coming to what is referred to as age pyramid. So by knowing the age of the individuals of a particular population, age pyramids can be generated. Age pyramids are actually the snapshots of the age structure of a population at some period in time, providing a picture of the relative sizes of different age groups in the population. For example, this is the age pyramid of India of the year 2017. So males and females you can see. So these are the number of individuals or the percentage of individuals which are present. So these individuals, the population pyramid set, it basically shows that there are a lot of young people which is there. This is a growing age of the population. It's more of a growing population, because you can see that the higher ages, the more than 80, 90, the number of males and females are very less. However, the youngs are very large in number. So even the middle generation is very large in number. So this is a growing population, as we suggest. look at the age pyramids of United States of America and Nigeria. So Nigeria is a very fast growing. This is a relatively slower growing population. Because a slower growing population, there is more accumulation of individuals in the middle ones, and there is a lesser of birth which is taking place, although death is again, there is more in number. This is a rapidly growing population. As you can see, the number of individuals that are born are too many. So the number of individuals, they are growing at very large pace. In United States of America, you can see it's more of a stabilized, age pyramid. So you can see the number of individuals are actually more or less constant. So it's said to be a very stabilized population. Neither it is increasing nor it is decreasing. So it's a very stable population, stable age structure as far as it is concerned. So this is a rapid growth. So these are the general examples. Those were specific. So the Guatemala, Nigeria, as we saw in Saudi Arabia, you can see that there are more of pre-reproductive or the juvenile individuals, less number here and less. So this is a very rapidly growing. This is somewhat what the India was also of like. So the Australia, Canada, India was somewhere in between here. So this is kind of a slow growth which we talk about. This is something which is zero growth. So in 2017, actually US is achieving this kind of a zero growth. The Spain, Austria, Greece, this is more or less a constant growth which is there. And this is something which is called as a negative growth. The Germany, Bulgaria, Sweden, there the old people are many more than the young people which are there. So that even the newborns are very less. The pre-reproductive individuals are less. So this is a negative growth population what we talk about in the age structure which is there. So we have talked about the population, the attributes of population, the one attribute of population which is basically the density, and that we talked about because we talked about the spatial distribution of individuals in a population. So spatial distribution or the spatial structure of a population as well as the age structure of a population we have covered. And that would be all for this particular session.
Thank you, ma'am. Thank you so much for giving deep insight into today's session. Friends, we believe that your feedbacks as well as your questions are important for us. So do write to us at info.cec@nic.in. We are going to meet again soon and would be discussing more. Till then, take care. Goodbye. Thank you, ma'am. Thank you so much.